Pleasure to introduce today's speaker, Dr. Melanie Medina López. I hope I did pronounce her name correctly. Dr. Medina López is a postdoctoral researcher at Penn State, where she works with the Department of Plant Pathology and Environmental Microbiology and the Department of Food Sciences. Originally from Puerto Rico, she earned from the Ohio State University, focusing on the interactions between fungal communities and plant parasitic nematodes. Her research explores the complex relationships between soil microbiomes, plant health, and beyond her scientific contributions, she's passionate about bioinformatics, science communication, and outreach. We are excited to have her here today to share her work. Please join me in welcoming Dr. Medina López.
Thank you so much for the introduction and good morning, everyone. Today, I'm just going to be sharing a portion of my PhD work with fungi and the soybean cyst nematode in Ohio soybean fields. And towards the end of this presentation, I'll be sharing a bit of the work that I'm just starting as I joined Penn State a little over a month ago, I would say. So, let's see if perfect. Unless you're a plant pathologist or a nematologist, you may not have heard about the soybean cyst nematode. So the soybean cyst nematode, or SCN for short, is the common name of Heterodera glycinus, which is a plant parasitic nematode that primarily infects soybean, specifically in the roots. This nematode is characterized by the formation of a cyst. As you can see in the figure here, the females of the nematode once their eggs are fertilized, they die. And as the body of the female hardens, it forms this shell around those nematode eggs, protecting them from environmental factors and desiccation. And this is one of the reasons why this nematode has become one of the most important yield-limiting pests of soybean in North America, because as those eggs are protected within that cyst, they can really stay in that soil for a long time. Currently, this nematode has been reported in over 82% of Ohio's counties, and in the figure here, you can see that it's really widespread across the US. So it's a pretty important problem for Ohio soybean growers and the rest of North America soybean growers. One of the challenging parts of this disease is that we have very limited management strategies. So there are no nematicides that you can use in the field at this level or at this scale of that we grow soybean, because nematicides can be very damaging to human health and environmental health. So we regulate those very tightly. So the growers only have two options: choose resistant to the nematode, and then establishing a very rotation where it usually lasts 6 years, and you rotate from resistant soybean varieties and non-host like corn. With that in mind, as part of my research, we're allies within fungal communities. So since we don't have many tools to manage the nematode, we were hoping to use fungi to attack soybean cyst nematode in the field. So we know that some fungi can attack nematodes in general. These fungi are called nematophagous fungi or predatory fungi, and they can attack nematode eggs, but they can also form very intricate traps to literally trap nematodes. These can be in the form of sticky traps, or as you can see in the figure here, the fungi can form specialized traps like this one that has a constrictive ring, where as the nematode goes into the ring, those cells that form that ring swell and literally strangle the nematode to death. And then the fungus uses that nematode as a food source. Some previous studies have produced successful antagonism of soybean cyst nematode and related nematode species using these group of fungi. And we were hoping to do some of that same work in Ohio. So with that in mind, we went into this research with two primary questions and objectives. The first was we wanted to know which fungi were already in those Ohio fields to see if any of them had any potential for biocontrol, but also to understand a little bit better the biogeography of fungi in agricultural systems in general. And we also wanted to learn if we could use any of the fungi that were already present in the fields to help us manage soybean cyst nematode. So to address that first question of which fungi were already within the state, we did a survey experiment that started in 2019, and because we all know what happened there, and we wrapped up sampling in 2021, where we would either visit farmers' fields that were contacted randomly, or they sent us or submitted samples to the Ohio State Plant Diagnostic Clinic, and from there we took a subsample to use in this study. In total, we collected 171 soil samples from different fields, and those field locations here. Once the soil was collected, we extracted DNA from the soil to identify the fungi within that soil sample using long-read amplifiers, encompass the full ITS region of the fungi. So that includes a portion of the 18S region, ITS1 and ITS2 regions. We counted the nematode eggs on that soil sample, and we tested the edaphic factors of the soil. And then we put all of that together to analyze the fungal communities of the soil and how soybean influence that fungal community. So going into our first finding was that soybean cyst nematode had higher abundance in the major soybean production regions of the state. So on the map here, the circles are colored blue depending on how high was the nematode abundance within that sample. So the darker the blue, the higher the nematode abundance. And if you look at the west region here, they have the highest level of soybean cyst nematode. And one of the reasons for this is that that is just the general area where we produce most soybean in the state. And as any plant pathogen, the more that you plant a susceptible soil host within it, accumulate that pathogen. As these growers have been continuously planting soybean in that field, the abundance of the nematode goes higher if the nematode is left unmanaged. So for the rest of this presentation, I'm going to be talking about soybean cyst nematode abundance in terms of categorical values, instead of using the specific number of soybean cyst nematode eggs that we found in the sample. To do so, we used as a guide these that we give growers in Ohio specifically, where once you have over 100 cc's of soil per 100 cc's of soil, then that's the point where you should start thinking about management of the soybean cyst nematode within that field. So we set that soybean cyst nematode abundance since it already warrants intervention, and then worked ourselves down from there. Okay, so now I want us to move into thinking about how the environment fungal communities of that soil. And we start there because we know that environmental factors and soil-related factors are gonna influence that fungal community. Want to have to keep that in mind as we think about the effect of the nematode later. To do so, we did a principal component analysis using the edaphic factors of the soil, which included the variables on this graph here. I'll just mention a few ones like pH. We looked at the sand content within that soil, things like organic matter content, total exchange capacity, etc. And we also included climatic factors like mean annual temperature and mean annual precipitation in our analysis to account for those environmental factors. And we found regional differences in the combination of edaphic factors and climatic factors that led us into drawing some regions within the map that would help us then understand what were the differences in fungal community composition that were associated with climatic factors of that region. And you can see those regions drawn here, and the colors correspond also to that principal component analysis. Okay. So the colors are still the same. So what we're looking at here are the results of a k-near neighbor analysis that analyzed how the fungal community composition of the soil was influenced by the geographic region, which just corresponds to those edapoclimatic factors. And what we fungal community composition was indeed influenced by edapoclimatic factors. And the way that we demonstrate this was by running this k-near neighbor analysis. In basically what a k-near neighbor analysis does is that it takes samples and compares them within each other and then selects what is the most closely, the most similar sample within the rest of your samples. So in this case, it would choose the fungal communities compositions that were closer to each other. And then this would be an iterative process where it would compare one sample to all other samples and run this multiple times. Then if the two selected as the most similar, it will draw a pure edge type. However, if through these iterations, different samples were selected as the most similar, it would draw a mixed edge type. So with that in mind, if you have a pure edge type, it means that if you ran that comparison 100 times, those two samples were selected as near neighbors. And what we found was that within this network, there were several modules that corresponded to regions that were all formed in pure edge types. Here, I've pointed out an example of a module for the central region. Here's one for the south region, and one example for the west region. So in general, this tells us that edapoclimatic factors and geographic regions plays a significant role in that fungal community composition. And that's not entirely surprising because we do know that environmental factors influence microbes in general. So we wanted to learn if soybean cyst nematode abundance had any correlation or influence in the fungal community composition, regardless of where that sample was collected from. What we did is that we performed a fungal indicator species analysis. An indicator species analysis just identifies species that are consistently found within the same sample group. So if a taxa is consistently found within the group in all of your samples, or a significant proportion of those samples, it would be identified as an indicator species analysis, indicator species for that group. And what we found is that regardless of the region, you would see some of those species popping up depending on the soybean cyst nematode abundance within that sample. In this case, we saw that if you had high levels of soybean cyst nematode abundance, then you would start to see a higher number of those indicator species analysis being the same. So you can see the soybean cyst nematode abundance, it's marked in different colors, and the high level is that orangey color. And all of those samples clustered within the center. However, the samples with all other levels of soybean cyst nematode abundance did not cluster significantly in any manner. So that tells us that once you have a high level of nematodes in that sample, there is some selective pressure starting to happen as a result of the nematode being there, or as a result of the pathogenic infection, or as a result of the changes that are happening in the plant exudates as a result of infection. We don't know exactly why, but at least this gives us enough evidence to continue studying leading to this change in the fungal community composition. So delving a little bit deeper into more of the specific of which are the fungi that are changing more significantly with these differences in soybean cyst nematode abundance, we ran a core community analysis, which basically just identifies shared community members within the different groups. And we used a prevalence of 75% and a minimum detection of 0.00 abundance in at least 90% of the samples. And before I go into talking about the taxonomy of some of this, I want to make clear that we use ITS sequencing for this for these analysis. So the taxonomic assignment for all of the fungi can determined just with ITS. More specifically, fungi like Fusarium need several genes to be able to appropriately assign to the species level. So the taxonomic assignments that you're going to be looking at here are all putative and need to be independently verified for us to be sure that these are the fungi that we're actually seeing. But what was interesting is that some putative soybean pathogens were found associated with soils infested with soybean cyst nematode. One particularly interesting one was Fusarium virguliforme. It's unfortunately that is a Fusarium because we cannot tell if it's exactly Fusarium virguliforme specifically, but we did find it associated with high levels of soybean cyst nematode. And this is interesting because these two pathogens, Fusarium virguliforme, which causes sudden death syndrome of and soybean cyst nematode, act synergistically usually, where they cause worse disease severity when they are together than they would have caused if you summed their damages individually. And even though we can't confirm that the one we're seeing in association with high soybean cyst nematode is definitively Fusarium virguliforme, we have been receiving increasing reports of sudden death syndrome of soybean in the plant disease diagnostic clinic at Ohio State. So that together with we seeing it as core members of the community at high levels of soybean cyst nematode does raise alarm bells that maybe it's something that we should be looking into further. Another interesting fungus that was associated with low levels of soybean cyst nematode abundance was Cornospora cassicola. And this is the causal agent for target spot of soybean. However, it hasn't been reported in Ohio yet. So we think that there's maybe two things happening here. Either this is a Cornospora cassicola that is not a plant pathogen and it's just the same species, or the conditions within the state are just not conducive to disease at this point. So it would still be interested to look more into the species to determine whether it can actually become a problem. We are seeing changes in climatic patterns that could eventually lead to conducive disease down the line. On the brighter side of things, we also found some putative nematode antagonists that were common across the soybean fields in Ohio. Both Fusarium ecrozeti and Clonostachys rosea had been previously identified as antagonists of nematode. Clonostachys rosea specifically has been reported many times because they are entomopathogenic. And usually when you have a fungus that is entomopathogenic, it can cause a level of predation or antagonism to nematodes too, since nematodes are closely related and have some components that are similar to insects. they're not insects. So these were some interesting and salient taxa, but we also wanted to know if there were any fungi that were more or less abundant, whether depending on whether you had high abundance of soybean cyst nematode or low abundance of soybean cyst nematode. So we did a differential abundance analysis where if you see here, we find the same two putative nematode antagonists I'd mentioned previously being more abundant when soybean cyst nematode is high. And these may seem a little counterintuitive because why would the fungi that are supposed to be antagonizing the nematode be more abundant when there is higher level of the nematode? Like, aren't they supposed to be fighting each other? And like the fungus is supposed to be killing out the nematodes? Well, it's complicated. And I want us to think about how suppressive soils are developed. And if you're not a plant pathologist, this may be a new concept for you. So suppressive soils are soils that reduce the level of disease in a field. So it can either do it by reducing the actual inoculum of the disease in a field, or it can do it enhancing the plant's immune response that leads to less damage of pathogens to the plant. So the way suppressive soils usually are developed is that you have an initial increase in disease. So that increase in disease leads to the selection of pathogen-specific antagonists within that community. And as those pathogen-specific antagonists increase in number, then they lead to disease suppression. But that disease suppression in the long term just decreases that selective pressure of pathogen-specific antagonists, and the cycle starts again. Usually, once you get to this point where you have a decrease in pathogen-specific antagonists, this effect can be pretty long-lasting, last decades within a field, but it can eventually revert into being conducive to disease. What we think we're seeing in our results is that we are capturing a snapshot of our community at this point, where we see that increase in disease pressure, and we start to see that selection of pathogen-specific antagonists. But because we're always trying to reduce the level of disease, once we have that high level of disease, we just never get to this point with our current management strategies. And that makes sense, right? We don't want to let a pathogen just run rampant through our field. We want to stop the disease, but it also stops us from building suppressive soils at a significant scale. But would be potentially happening, and that this selection of specific pathogen, specific antagonists to those pathogens may be happening. So what can we do with what we have learned? Can we target specific field locations for fungal bioprospecting with what we have learned? So if we were to go back into the field and look at the locations that have that very high disease pressure, are we going to have an easier time finding those antagonists in the field? So that's exactly what we did. So we had a project with a grower that just let us go into his field and grid sample in his field. I have the fungal community data for these fields, but I'm not going to be able to share them today because of time. But basically, we went in and we selected field locations within these growers' fields that had the highest pressure of soybean cyst nematode. And we performed a high throughput isolation of fungi and tested those fungi for their biocontrol potential. Because we had such a large culture collection by the end of this project, we needed a way of filtering through those fungi to find the ones that had the highest likelihood of being effective against the nematode. So we started by running some in vitro assays. So we just plated the fungi amended with either chitin or proteins. In this case, we use skim milk agar. And then we challenged the nematodes with the fungus to see if there was any effect. And here you can see sort of the pipeline that we went through to test these fungi. I want to give a special shout out to Aranaya Cohen, which was the undergrad that did a lot of the heavy lifting to get this isolate collection up and running. In the end, we were able to collect 619 isolates, and those isolates were grouped in their microscopic morphology into 264 morpho groups, and we randomly selected a morpho type from those groups. Unfortunately, 25 of them did not survive our long-term storage methods, so they were unrecoverable after we had stored them. But of the tested isolates, 239 then went in to be tested for protease activity, extracellular protease activity, in that plating assay that I was telling you. Then anything that was positive for protease was also tested for chitinase activity. And you may be asking yourself, why choosing these two specific assays for? The reason is because the nematode eggs, they are made of different components. And as we know, proteins are really what make up everything within our cells. But chitin specifically, it's very important in the nematode eggs because it protects the eggs from desiccation. So if you see here, it's one of those first layers of protection of the nematode inside. So in red, you can see the chitin layer. And chitin, it's a very strong component of that eggshell. So we were thinking, well, if it can produce proteases and it can produce extracellular chitinases, then it has a higher likelihood of being able to chip through that egg and get at the nematode inside. That was our thinking here. So once we had tested for protease and chitinase, 71 of those fungi were actually positive for the production of both of these extracellular enzymes. Then we started looking at how these fungi grew in the lab, because the methods that we developed to challenge the nematode with the fungi to working with fungi that had yeast-like growth at room temperature. So we separated the fungi into everything that had yeast-like growth and everything that had filamentous growth after, so that we could start developing different approaches to test anything that had yeast-like growth, and we could continue to test the things that had filamentous growth at room temperature. Okay, so then after we had filtered for the things that may have the highest likelihood of going through that nematode eggshell, we ran an soybean cyst nematode egg colonization assay. And the way that that looks like, as you can see in the figure here, is that basically you're gonna put the fungus in the center of a plate, and then this nematode cyst. So if you remember that female nematode with all the eggs inside, you're gonna surface sterilize those and plate them around a circle. You allow that fungus, and then you look under the microscope of whether the egg was colonized or not colonized by the fungus. And again, it might be hard to picture how you would be able to see the fungus within the nematode egg. So I have here an example of how that would look like under the microscope. So if you have a healthy soybean cyst nematode egg and the nematode egg is mature, the eggshell becomes really transparent as it basically stretches to accommodate for the growth of the nematode. So you should be able to see the nematode folded in there and all of its parts. It's beautiful. And then if the nematode egg is colonized by the fungus, well, then you don't see anything. Now you just see a fungus. So this is an example of one of our isolates. This is isolate NC423 here in the graph. And what I'm showing in the graph is the percentage of eggs that were colonized in our test. And we found that 5 of our fungal isolates significantly colonized soybean cyst nematode eggs in this assay. But we know that colonization alone, it's not the only strategy that these fungi can use to predate or parasitize nematodes or eat them. So we used a different approach to see if we can find any potentially nematophagous or nematode antagonistic fungi that we may have missed in the using the previous assay, because they just have a different approach to antagonizing nematodes. In this case, we ran egg hatching and motility assay, where same thing, we plated the nematodes alongside the fungus, and then we crushed those cysts and we put them in a hatching chamber. A hatching chamber is basically a chamber that has a sink and water solution at the bottom, and then it has a thin mesh where it just comes into light contact with the eggs. So you can see an example of how our actual setup looked like here. This is the fine mesh, and then you would have that sink and water solution at the bottom. So any nematodes that hatch and were still motile would tend to swim through the mesh and get to the bottom where the rest of the water would be, because they avoid desiccation. So they just avoid the air, basically. So then when you want to remove that mesh and get it to the water at the bottom. And what we found was that 8 of the fungi that we tested significantly reduced hatching and post-hatch motility of soybean cyst nematode. And interestingly, if you remember, previously we had only found 5 fungal isolates that were visited by the fungi and can reduce hatching and post-hatching motility, which just highlights the importance of using different methods for bioprospecting, because if you just use one, you may be missing some microbes that have a good potential for being the pathogen that you're testing, but they're just not all assays will be good for testing different methods of antagonism. So by this point, I hope that I have shown a little bit of which fungi were already there in those fields in Ohio soybean soil. So we were able to characterize some of the fungal community composition in Ohio soybean field soils at different levels of soybean cyst nematode infestation. And that we evaluated the antagonism potential of soil fungi from Ohio soybean fields against the nematode. And we're very excited about the second part, because out of all of the fungi that we found at least one novel species that had never been reported as being antagonistic against nematodes at all. So we are very hopeful that once we go through some additional tests, we'll be able to put that name out there. Maybe people can do some other cool research with this specific fungus. Now, moving into my work here at Penn State. So I just recently joined the Dr. Bull Lab and to study not only fungi that are predatory, but really because fungi is just one example of the multiple micropredators that you can find in the soil. So I've been given the opportunity to continue to study micropredators just from a wider scope. On titled Hunting for Healthy Soils, Organic Management of Soil Micropredators for Pathogen Control. So as I said, there are many other microorganisms that also lead predatory lifestyles. And as you saw an example for just now, these can consume plant pathogens and human pathogens. So this project that I've just joined really goes at the overarching question of how can we harness these micropredators to support plants. And to do so, this has been breaking down into several different questions that tackle this problem specifically using as a system because organic farmers have like specific challenges when it comes to the management of plant pathogens because they have limited management strategies. So looking at the potential of micropredators to support plant and soil health within this system is very beneficial. So we want to learn about which features of organic soil management and amendments can influence micropredator communities. We want to learn about what effects soil organic management amendments have on these communities and their capacity or effectiveness to suppress pathogens. We also want to identify micropredator groups that would be most beneficial with plant health. And we want to know how we can best apply and share this knowledge for the benefit of these organic growers. And at Penn State specifically, we'll be applying several approaches. The first of which is a survey of organic farm soils that will be conducted in several states, including Pennsylvania, of course, Connecticut, California, and Florida, where soil from different organic farms will be collected, and we'll look at what the management approaches are in these farms, and we'll hopefully get a better understanding of what the general predator communities within these organic soils is, and then how management practices and amendments may influence these micropredator communities. We will also look at the effect of organic amendments directly in the predator performance against pathogens. So we would do this in a series of microcosm experiments where different organic amendments alongside different combinations of micropredators and plant pathogens to hopefully see if any of these organic amendments best support the performance of these micropredators to suppress disease of plants. And then we'll engage in several extension activities like webinars, Q&A, writing some fact sheets so that we can communicate the results of this research and any important findings for management to the growers so that we are actually putting into practice what we learned through this research. And I'll just take a moment to acknowledge everyone that was a part of my PhD experience on the left, and then the new team that I just joined here on the right. I lost the logo right here. The CFAS, Ohio CFAS was right here. The Ohio Soybean Council, which funded all of my research and my PhD, and Penn State and USDA NEFA, which fund what I'll be doing right now. And with that, I will take your questions.
By tradition, we take our first question from a trainee. Any questions?
So great talk, by the way. I learned so much. So I don't work with fungi, so I'm not like used to what kind of genomic tools maybe you use for ITS, and do you face any issues in terms of coverage because fungi genomes are much larger? So does that lead to any issues or like any advanced, like do you need, what additional sequencing would you need to do on top of ITS to confirm to species level?
So that would depend on the specific group that you were wanted to target if you're gonna keep using amplicon sequencing. So with fungi, that's really one of the main drawbacks of using amplicon sequencing. A lot of these um, species are very complex in their taxonomy. So you would need several other genes to accompany ITS to be able to appropriately assign taxonomy to these. So if you were to do a like metagenome approach, that would help out a lot. The problem is that when you're working with soils, using metagenomic approach is very challenging because there's just so much other eukaryotic DNA there that a lot of your sequencing gets eaten up by off-target organisms that you're not necessarily looking for. Moreover, since fungi are eukaryotes, you're also gonna sequence, even if you just do like a targeted approach of you just use this ITS sequencing, you still get off-target amplification. So, I mean, for now, ITS is just the most cost-effective way in which you can identify the fungal communities within the soil. You could use shotgun, but you have to be prepared to just lose a lot of your, I don't want to say lose because it's still data, but just sequence a lot of off-target organisms. Some people use enrichment approaches where you enrich for your target microorganisms and then sequence, but that introduces additional variation. So really it's a matter of what's your question and then choosing the best approach to answer your specific question. Our question was generally trying to identify what was there because we knew very little. So the approach worked well for our specific question. I don't know.
Hey Melanie, that was an awesome talk. I was curious about, so do the nematodes have some ways of evading the predation from the fungi? And if they do, is there a genetic basis for it that could be selected for over time?
So yes, but it's the fungi have very different approaches to how they are predaceous to the nematode. Some of them will have these physical traps, and nematodes have developed evasion measures for those physical traps. There's this super cool study that basically touched the back of the head of nematodes, and nematodes have this reflex where they just swim back. And they think that this is like an evolutionary arms race between the nematodes and nematode trapping fungi, where if the nematodes are able to swim back before those traps close, then they may be able to evade that trap altogether. Also, in terms of sun, they can also thrash. So if you have sticky traps, the nematodes sort of thrash around and try to free themselves from those, swim away. But also nematodes have like immune responses to toxins that these fungi produce, so they can detoxify from some of the nematotoxic compounds that are produced by the fungi. So there's like a whole host of different strategies that nematodes use to defend themselves against the fungi. But as the nematode develops those, the fungi also develop better ways of attacking the nematodes. So it's arms race, right? We, at least as plant pathologists, are keenly aware of these effects of like, you have a great antibiotic and then the bacteria becomes resistant. You have a great antifungal, and then the fungus becomes resistant. The same way happens with nematodes too. I hand first.
Yeah, so I was wondering, so the goal is like to find like a fungi that can be used to like fight against the nematodes. So like how would it be like added to the soil? Like
That's a great question, because ideally, so if you are putting your industrial microbiology hat on, I take that microbe, identify what it is that it's using, and synthesize it either through bioreactor, or you work with a chemist and get it synthesized so that you have a molecule, right, that you can use as an active ingredient in whatever it is that you're using. The unique problem with nematodes, and why a lot of the compounds that we have found, because there's no shortage of nematotoxic compounds out there that we have identified. The problem is that nematodes are too close to humans, but not quite humans, and then too close to insects, but not quite insects. So if you try to attack them through those portions of their system that are too close to humans, then you end up harming humans. If you go the other side, then you end up harming insects. So it has been very difficult for us to find chemistries that are safe for us to apply in like a fungicide sense, where you can just go in the field, apply it, and have it persist low, like have low persistent in the environment and still have it be effective. We haven't found that yet. So the other option is putting the actual fungus in the field and hoping that it grows enough and it stays there enough to actually lower nematode abundances. And biocontrols in general, we find that they then you go to the greenhouse and they have modest results, and then you go to the field and 1 out of 10 fields you may see a significant difference. So we have problems of having these solutions or these microbes have consistent performance across fields. So our hope was that if we the same place that we have the problem, it may be better adapted to that environment. So with a little tweak here and there, we may be able to have it stay there long enough that it has an impact. So that's our hope. The science is just not there yet, which is why we need a lot more. First of all, learning about the ecology of these microbes would be very helpful to see really what is it about the environment that allows this microorganism to persist better in a field. And then we just need a lot of help from like engineering better microbes that can resist and persist in the field. So our hope is that we can put it in and it reduces the nematode, but we still have not been able to find consistent ways to do that. I think I don't know.
I guess kind of a little bit more on that management side of things. Maybe instead of like adding the microbe, could we cultivate it within the field? And I know you mentioned that there was like a 6-year management practice, a 6-year rotation. I guess I'm wondering, how well does that align with maybe practices that could cultivate this microbe in the field? Like, is that 6-year management practice, is it cultivating these microbes and that's why it's reducing soybean cyst nematode, or is it something else that is maybe disrupting that pest and disease cycle?
So nematodes in general, once you have it in the field and you have a good host, it's just gonna stay there. You're not gonna be able to get rid of it. The problem with cyst nematodes is that you can have a cyst nematode there for 6-7 years with no good host, and it'll come back. So management wise, other than diversifying your production, there's not much that a grower can do to reduce the levels of the nematode because it can just persist there that long. And the reality of our farming system is that it will cut deep into the pockets of farmers if we were to tell them, well, you know, you have a nematode problem, you need to grow something else. Like, they've already invested in all of the tools. They buy seed with, like, if you are a soybean farmer, by December, you already have your seed order out. So you need to know with a lot of time in advance, what is it that you're gonna plant in your next growing season, and changing from soybean to a different crop it's a significant investment. That's why when you look at farms in the Midwest specifically, you're gonna find a lot of soybean, a lot of corn, because you can rotate between those two without major changes to your operation. So it's not feasible to ask these growers to significantly change their management practices unless you can tell them, yes, this for sure will help your yields. So, and as a plant pathologist, I wouldn't feel comfortable going to a grower and saying, invest however many dollars and see if it works, because they need to make a living. So if we're gonna make different management recommendations, we better be damn sure that they're gonna pay themselves when the grower implements them. So I think that's the major challenge there.
Thank you. Great talk, Melanie. I'm just curious if you observe any variation in the abundance of the fungi throughout a growing season, for example, from planting to harvest, because I'm thinking about if these observed effects that you showed us are permanent or they are temporary.
So I can tell you for the core community that we identified, that core community is stable throughout. Because these samples were not collected during the same time period. These samples were collected across a field. If you remember that field experiment that I showed where we got isolates from those fields, that was a 2-year field experiment where we sampled at the beginning, in the middle, and at the end of the season. And we looked at the fungal community composition of each of the individual grids. And we found a lot of the same core community members of fungi that we found in that survey, also as part of the core community members in that field that was not included in the survey. So there's like an independent sort of testing of whether what we had found during the survey was consistent with different fields that were not included. But we do know changes through time, especially according to the developmental stage of the soybean. So when the soybean is in its vegetative state, so that's before they put out any flower, you're gonna have a distinct fungal community composition. Once it gets into the reproductive stage, so once it starts flowering, you're gonna see a distinct community composition. And then when it starts filling pod, that's a very highly metabolically active plant, it's also gonna have a different community composition. So sampling point is important, but there seem to be a few group of microbes that are consistently present within the field if you have soybean growing there.
We have time for one more question.
I think back there they had their hand up a few times. I don't know if you want to ask.
I forgot. It's okay.
So in your study field with low nematode population, do you by any chance find penicillin species?
Fungi?
Yes.
Yes. So during the isolation of fungi and during the during the isolation and during the barcoding approach, we did find pennies, but significantly, they didn't have like any significant differences in between the different levels of soybean cyst nematode, and they also were not very effective at producing extracellular enzymes when we tested them in vitro. So they're in our culture collection, but we haven't really found any salient activity of those.
I know it has been biologically used to control.
But it's possible that, so some of these nematode antagonistic and predatory fungi, they do exhibit a little bit of preferences. So there's like a little bit of niche differentiation depending on their strategy. So if Penicillium is a primarily producing nematotoxic compounds, it's possible that it's not going to produce any unless it senses nematodes in the environment, because a lot of these fungi do turn on and off their nematotoxic production depending on whether they sense nematodes around or not. So that's another thing that we were thinking about as an our collection again, but testing them always in the presence of the nematode, because that may be influential in our results eventually.
All right, thank you everyone for joining us for this